In this class, we are going to study about the third category of the kingdom Protista, and that is the Euglenoids. So just like the previous two categories, that is the chrysophytes and also the dinoflagellates, this category is also mostly photosynthetic. So all these three categories, that is the chrysophytes, dinoflagellates and here the euglenoids. So these three categories come under the Protestan alga, and obviously this means that this category also performs nutrition or obtains its nutrition mostly by photosynthesis. So it's not that only photosynthetic organisms are present over here, some other sorts of nutrition are also present, but mostly these organisms are photosynthetic. And obviously if we are studying the kingdom Protista, we know that these categories will be unicellular. So photosynthetic and unicellular are the first characteristic of this category, and also these are motile. So this is what making it different from the chrysophytes. So we had seen that in chrysophytes flagella was normally absent, and therefore the chrysophytes were floating passively, they cannot perform active locomotion. But these are flagellated, that is these organisms do possess flagella and therefore these are motile. And we know that flagella is normally used in aquatic medium. So obviously if the presence of flagella is over here, this means that these organisms will live in aquatic environment, be it fresh water or be it marine water, and also in very moist condition like marshy soil or very moist soil or muds, puddles, ponds or anything of that sort. So obviously when flagella is present, it means that that organism is normally living under moist or the aquatic conditions. So these are flagellated and motile. And there is a very important feature over here, and that is cell wall is normally absent. So in our previous two lectures we had studied about chrysophytes and dinoflagellates, and we had seen that cell wall was present over there, but here cell wall is normally absent, but there is a covering instead of the cell wall. It's not that cell wall is absent and nothing is there at its place. Obviously there is something, and that is called a proteinaceous layer, and that is named as pellicle. So instead of cell wall, here pellicle is present. So pellicle is normally composed of proteins with a very small amount of lipids and also a few carbohydrates. So this is the characteristic of these organisms, that is these are unicellular, and apart from that these are photosynthetic, and obviously after that these organisms do not have cell wall but have pellicle in presence of that, and obviously these are flagellated and motile. So this line tells us about the basic features or properties of these organisms. Apart from that, we see that pellicle is sometimes rigid. So normally pellicle makes the cell flexible or elastic. So normally when cell wall is present, it makes the outer area of the cell rigid, or as a whole the cell wall makes a cell rigid. But in this case pellicle will make the cell flexible, and we will later see that why there is something called myonemes and also some other structures are present. So this pellicle normally makes the cell flexible, but the cell wall makes the cell rigid. But sometimes pellicle makes the cell rigid, but normally when it is asked that whether pellicle makes the cell flexible or rigid, so we must understand it makes the cell normally flexible. Sometimes a few exceptions are there where pellicle is rigid, or it is giving cell wall like property, that is rigidness to the cell. Now pellicle normally composed of proteins with a small amount of lipids and a very small amount of carbohydrate. So the amount of protein is so much in this case that we can assume, or we can say that the pellicle is a proteinaceous structure. We don't need to say, because the lipids and carbohydrate content of the pellicle is so low that we normally consider it as a proteinaceous structure. So even if we say that only proteins are being constituting this pellicle layer, then it is also almost correct, because the pellicle is normally mostly composed of proteins. Now in our previous class we studied about dinoflagellates, and we saw that dinoflagellates have two flagella. One was transverse and the other one was longitudinal. Here also two flagella are present, but these are not transverse or longitudinal. One flagella is short and the other flagella is longer. Now the shorter flagella is attached to the longer flagella in a region which is called as paraflagellar body, and that paraflagellar body is also a photosensitive area. So these are the two flagella, a long and a short one. The short flagella is attached to the long flagella in a region which is called as paraflagellar body, and that is also an area of photosensitivity. So that area can detect light. So this is the difference between flagella of euglenoids, that is one short and a long one, and in dinoflagellates also two flagella are present. So one was transverse and the other one was longitudinal. Now beneath the layer of pellicle, we know that a cell has cell membrane, and after that if something is present, normally cell wall. Then beneath the cell wall, because here we don't have a cell wall, we have pellicle. So beneath the pellicle there are some structures which are contractile in nature, therefore they can make the cell contract. The body of the euglena can contract because of certain structure which are called as myonemes. Now these myonemes are present in various rows or in various designs beneath the pellicle, and there is something called metaboly. So metaboly is a sort of wriggling motion which occurs in the case of euglenoids. So for that wriggling motion to occur, for that wave of contraction and relaxation to occur, these myonemes are very important. So obviously flagella does perform locomotion in the case of euglenoids, but also there is another type of movement that is called as metaboly, and metaboly is mainly because of these myonemes, because metaboly involves contraction, a wave of contraction and relaxation, and it is because of these myonemes, because these are contractile structures, so these can make the euglena contract and relax. So therefore these are also a sort of locomotory helper organs or structures. Now mucilage is also present. So in our other category we had seen mucilage propulsion. So mucilage propulsion was helping in the gliding motion of the chrysophytes, but here this mucilage is not for that purpose. Here mucilage has a different purpose, and that is prevention of the cell from losing its water content or desiccation. So when the cell undergoes drying condition or the environment is not suitable enough or moist enough for the euglenoids, then the euglena secretes a layer of mucilage around itself, and that mucilage prevents the moisture of the euglena from, or the euglenoids from, escaping. So this mucilage is normally released or secreted under desiccation conditions and also during cyst formation. So when the adverse environmental conditions last for too long and the euglenoids cannot survive that condition, in that case cyst wall formation takes place. And for the cyst wall formation to take place, mucilage secretion is a must, and therefore mucilage is secreted either during desiccation or during the cyst wall formation. So both of these conditions are adverse environmental conditions for the euglena. So this desiccation and the cyst wall formation, these are the two main times during which the mucilage secretion occurs. Now there is something called as Osmo Regulation. So Osmo Regulation is the regulation of fluid and ions or salts in that. So Osmo Regulation almost takes place in all organisms. So Osmo Regulation here takes place with a structure called vacuole, and in vacuole there is a special type of vacuole which is present over here, and that is called as a contractile vacuole. So Osmo Regulation is the regulation of the normal water content and the ions or the salt content, and that is being done with the help of the contractile vacuoles. So in humans, in our body or in other mammals, this osmoregulation occurs through the help of kidneys. Our kidneys regulate the amount of water to be excreted or the urine to be excreted, and also in urine the various salts are present. So here the work of kidney or something like kidney is being done with the help of contractile vacuoles. Now there is another structure which is photosensitive, and that structure is called as the eye spot. So we know what is the purpose of our eyes. The purpose of our eyes is to detect light and visualize the environment. So here also there is something called eye spot. So it is not exactly like the eye, but still it is photosensitive. Our eyes are also photosensitive. This is also photosensitive, and it is because of the presence of red carotenoids. So the eye spot it contains reddish granules of carotenoid pigment. So this is the pigment which provides photosensitivity. That is the ability to detect the light is called as photosensitivity. So there are two parts of photosensitive areas, and one is the eye spot, and the other photosensitive area is the paraflagellar body. And where is that? Paraflagellar body is the point where the short flagella is attached to the long flagella. So the paraflagellar body and also this eye spot, these two are photosensitive areas in the case of euglena. So these help in light detection. And why it needs to detect light? Because obviously these are mostly photosynthetic. So for photosynthesis to occur, light is a must, and the plant or the euglena has to be able to detect that light, and also because of some other conditions we will see that. Now we have studied in the first line, these are photosynthetic. So for photosynthesis to occur, it is very much obvious that pigments like chlorophyll should be there. So here there is a very interesting thing, and that is chlorophyll B. We know chlorophyll A is almost everywhere. So here also chlorophyll A is present, carotenoids are present. So we had seen that chlorophyll A and carotenoids were present in chrysophytes and also the dinoflagellates. We had seen that in our previous classes. But here this chlorophyll B is coming. Now this chlorophyll B is important here because this pigment is also found in higher plants. Obviously these chlorophyll A and other carotenoids and xanthophylls are present in almost all the photosynthetic organisms, but this chlorophyll B is mostly present in higher plants like angiosperms or the gymnosperms. So this chlorophyll B is important. That is why euglena might be considered as the ancestor of the higher plants, because the pigment that is chlorophyll B is present over here, and it is the same pigment which is present in the higher plants like gymnosperm or angiosperms. So that is why it could be considered as the ancestor of higher plants. Now we have seen that most of these are photosynthetic. So obviously photosynthesis is a method of autotrophic nutrition. So we can say that most of the euglenoids are autotrophic or performing autotrophic nutrition. So this is the most common method, but photosynthesis can only occur if the presence of pigment is over there. So only pigmented euglenoids are photosynthetic. Which are the pigments? Chlorophyll A, B and carotenoids and some xanthophylls also. So where the pigments are not present, over there the photosynthesis cannot occur, because the pigment is a must for photosynthesis to occur, and therefore non-pigmented euglenoids will be saprotrophic, that is they would be consuming dead and decaying organisms, and also parasitic. Parasitic means they would harm the host on which they live or inside which they live. So these two forms, that is heterotrophic forms. So saprotrophic and parasitic, both are heterotrophic modes of nutrition. So we see that mostly autotrophic nutrition is occurring where pigments are present. Non-pigmented euglenoids are heterotrophic. And what are the types of heterotrophic? So that is saprotrophic, that is feeding on dead and decaying matter of organic nature, or parasitic, that is it harms the host on which or inside which it lives. Now these are normally present in different organisms, but we have to study about a specific example, and that is Euglena. In Euglena there is presence of photosynthetic pigment. So Euglena is photosynthetic, that is Euglena shows autotrophic nutrition. But when the Euglena does not receive sunlight for some time, then it cannot synthesize its own food. So in that case it becomes predatory and it feeds on smaller microorganisms. So Euglena is autotrophic because it has photosynthetic pigment and it performs photosynthesis. Now Euglena is also heterotrophic, and it is predatory type of heterotrophic when it is deprived of sunlight. So when an organism shows autotrophic mode of nutrition and also heterotrophic mode of nutrition, in that case those type of nutrition is called as mixotropic. So this is a special type of nutrition which is between autotrophic and heterotrophic. So there is autotrophic where only autotrophic or photosynthetic nutrition takes place, and there is heterotrophic where only the nutrition is obtained from some other organism. But this is mixotropic. Mixotropic means sometimes autotrophic, sometimes heterotrophic. So this Euglena that we have to study in according to our NCERT, this Euglena is performing photosynthesis when it is receiving sunlight. So when it is not receiving sunlight, it cannot do photosynthesis because sunlight is a must for that. In that case, this Euglena becomes a heterotrophic and it becomes a predator and feeds on smaller organisms, and therefore this type of nutrition is called as mixotrophic. So according to our NCERT NEET syllabus, this is what we have to study and remember about the protist. And obviously the other properties will be same as the other category, that is binary fission would be the common mode of reproduction in this case, and also the other type of spore formation might occur sometimes, but normally binary fission would take place, and the rest of the properties are in general to all the protista, that is these are eukaryotic cells. These have normal mitosis method of reproduction, and also there are few other features. So these features are common in all the categories of kingdom Protista. So those features cannot be or need not be repeated in every category. So whatever were the important features regarding the euglenoids according to our syllabus has been discussed here. So now we have completed 3 categories, that is the chrysophytes, dinoflagellates, and here the euglenoids. Now only 2 categories are remaining, that is the slime molds and the protozoa, which we are going to study now.